Assalamu alaikum students. Today now we will discuss about tricyclic acid cycle. This is also called citric acid cycle, TCA cycle, or tricarboxylic acid cycle. This is also called Krebs cycle as well. TCA cycle essentially involves the oxidation of acetyl coenzyme A to carbon dioxide and water. This cycle is the central metabolic pathway for the respiration process mainly. The TCA cycle is the final common oxidative pathway for carbohydrates, fats, and amino acids. Talking about the history of the Krebs cycle or TCA cycle, a biochemist named Hans Adolf Krebs, who was born in Germany, worked in Britain. His discovery in 1937 of the Krebs cycle of chemical reactions was critical to the understanding of cell metabolism and earned him the 1953 Nobel Prize for Physiology or Medicine. In eukaryotes, he told that in eukaryotes, the reactions of the citric acid cycle take place inside mitochondria. Mitochondria is this kind of structure, and it is having matrix inside. Inner mitochondrial membrane is there, outer mitochondrial membrane is here, and all the reactions of Krebs cycle are taking place inside the mitochondria. TCA cycle supplies energy and also provides many intermediates required for the synthesis of amino acids, glucose, and hemoglobin, etc. TCA cycle is the most important central pathway connecting almost all the individual metabolic pathways. You remember in the topic of the glycolysis, we talked about the synthesis of pyruvate molecules from glucose, starting from glucose, and intermediately was glucose 6-phosphate. And we will be taking use of those pyruvate molecules during the TCA cycle. Reactions of TCA cycle involve oxidative decarboxylation of pyruvate, which is actually coming from the glucose, to acetyl coenzyme A by PDH complex. We will talk about these all reagents, reactants, and products in detail. And this step is called connect or linking step between glycolysis and TCA cycle. From pyruvate, here you see, from pyruvate, in the presence of NAD plus, which is actually reduced to NADH, and H plus is also available there. Carbon dioxide is released. In the presence of PDH, it is converted into acetyl coenzyme A. This pyruvate is converted into acetyl coenzyme A. This pyruvate is actually coming from the glycolysis part from glucose 6-phosphate, which was originating from glucose, if you remember the last slides of the glycolysis part. And glucose is 6-membered carbon, pyruvate is 3-membered carbon, acetyl is kind of 2-membered carbon because one carbon is released here. And this is 2-pyruvate. This can be multiplied by 2, this whole process, because 2 molecules of pyruvate are there from glucose. So acetyl, it links with the coenzyme A, and this is called acetyl coenzyme A, and then it enters into the tricyclic acid cycle as such, as it is clearly shown. It in the presence of citrate synthase, it means from acetyl coenzyme A there will be the production of synthesis of citrate. From citrate, in the presence of enzyme aconitase, water will be removed and cis aconitase will be produced. Again, in the presence of aconitase, again one water molecule is released and isocitrate is produced. Isocitrate will be converted into oxalosuccinate in the presence of NAD+, which is again converted into NADH, and these are the enzymes. ICDH, we will study the names as well during the next slides, and we will discuss all the steps in detail during the next slides. All these steps will move this way. The final product is oxaloacetate, and again oxaloacetate, it binds with acetyl coenzyme A in the presence of citrate synthesis to produce citrate. Let's talk about the details of the steps, and again we will touch this cycle. Step 1 is the formation of citrate. Which citrate? Actually this one. Let's see. Oxaloacetate condenses with acetyl coenzyme A to form citrate, catalyzed by enzyme citrate synthase, and this reaction is inhibited by ATP, NADH, citrate, competitive inhibitor of, these are the competitive inhibitors for oxaloacetate. This reaction is having resistance from ATP, NADH, or citrate, these moieties. So, in the presence of oxaloacetate and citrate synthase enzyme, acetyl coenzyme A, it reacts with this one and it synthesizes citrate molecule. Step 2 and 3, citrate is isomerized to isocitrate. Citrate is isomerized to isocitrate by the enzyme aconitase. This is achieved in a two-stage reaction of dehydration. This one is dehydration. First step of dehydration, second step of dehydration, and citrate is converted into isocitrate through the formation of an intermediate cis aconitase.